creativity, it can improve your sleep, and it can actually treat major depression, which is one of the most common psychiatric diseases. And I really loved being able to have that kind of impact on real people and help them understand why they're sick, help them understand how their diseases can be treated. And so I changed my major to biology. And then kind of as I took more classes, I realized that I was really interested in genetics, and that's why I decided to study it in grad school. So I think that you guys are probably familiar with some of this. This is just the basics of genetic disease and inheritance. So you might have heard of Mendel's peas, where he had, parental peas that have one copy of the yellow form of the gene and one copy of the green form of the gene, big Y and yellow Y, big Y and little y. But those offspring peas that have two copies of the little y instead of being yellow are actually green. And in a similar sense, here we have two parents that each have a copy of that little y that codes for some sort of disease, some sort of form of the gene that doesn't work. And so 3 out of 4 of their children will be totally healthy because they only need one copy of that gene that actually works. But their last kid, 1 out of 4 of their children, will get those non-functional copies of the gene, and they will get sick. So let's talk about some fun facts about genetics. You can just type into the chat, how many pairs of chromosomes does a human have? I don't think I can see the chat.
When you're presenting, it's difficult to see the chat, but I have multiple students saying 23. A couple said 46, so the 23 pairs of 46.
Okay, yeah, I found the chat. That's okay. Exactly right. You have 23 pairs of chromosomes or 46. How many genes do you think are in the human genome? You can just guess. You don't, just give me a number, throw something out.
You can speak too if you like, right? Like you can say it if you don't want to type it, it's up to you.
Yeah, these are great. Googleplex, that's funny. Yeah, so we currently think there are about 25,000 genes in the human genome, so it's incredibly complex. How much of your DNA do you think you share with everyone in your class? Yeah, this is great. All humans share about 99.999% of the same genetic material, which makes it really crazy that we all look so different and act so different. That's one of my favorite things. And in fact, we share 97% of our genetic material with mice and even 60% with bananas. Really crazy stuff. Now let's talk about rare diseases. So about 70% of all rare diseases have genetic causes. How many people in the United States do you think have a rare disease? Really a lot, even though they're rare? Yeah, these are really great guesses. This is way better than I knew. I was surprised when I realized that about 30 million people in the United States have a rare disease. And that's about 5 or 10% of the population, which is really crazy. And that's why it's so important that we understand how rare diseases are caused and how we can diagnose them and how we can learn how to treat them. So how do mutations cause disease? I think it's first important that we just go over what we call the central dogma of biology, which is that you have genes, that's your DNA. And that DNA is transcribed into like an intermediate form called RNA, which is then translated into proteins. And it's these proteins that really function in your body. They build your body. They are what, do all of the jobs that need to be done to keep you alive and healthy. And so we can kind of think of the human genome as a recipe book, like a really great recipe book with 20,000 recipes and really detailed instructions on how to make all of those recipes, or proteins in this case. And so let's say there's a problem in that recipe book. And instead of saying, add 1 egg to your chocolate chip cookie dough, it says add 10 eggs. Once you get down to that protein, instead of getting great chewy chocolate chip cookies, you're going to get like baked eggs, essentially. And that essentially means that this protein isn't going to work and you're going to get a disease. And right now, scientists know a lot about how we can sequence this DNA and figure out what are the effects on this protein going to be, but not always. And in fact, when patients that doctors suspect to have a genetic disease go and get their genomes sequenced, only about 50% of them are able to get a genetic diagnosis. The other 50%, we don't have a good idea of what's going on. They have all these mutations, but we don't know how significant they are. There's still a lot of work to be done in how we understand genetic mutations. So in my research, I'm focusing on glycogen storage disease. Glycogen storage disease. So glycogen is one of the most important sources of energy in the body. So you might have heard of glucose, which is essentially sugar, and that's what your cells use to carry out all of the functions that they need to. Glycogen is how that glucose is stored in your cells, so that when that glycogen is broken down by one of these proteins over here, it can immediately be used as an energy source. So let's say you're running a marathon, you'll start to break down that glycogen. The glucose will enter your bloodstream and go to your muscles and all of the cells that need energy so that you can keep running and running and running. And if you have a mutation in one of these several genes, if you have 2 recessive mutations in any of these genes, you're going to get a glycogen storage disease. And depending on which gene those mutations are in, it can cause a variety of symptoms. So the most common one is what doctors call failure to thrive, which essentially means that babies and children just grow really slowly. They don't really hit the developmental milestones on time. They don't start walking and talking. They're generally a little bit sickly. Other symptoms are, really weak muscles, which makes sense if they can't get the energy they need. They won't have good blood sugar control. So if you can't break down this glycogen, well, that means that your blood sugar is just going to go up and down, and it's just incredibly bad for all of your organs, incredibly bad for your cells' ability to get energy when they need it and when they expect it. And that can cause really serious symptoms like heart arrhythmias. It can cause your liver to swell up and fail, and sometimes you'll even die. My friend is working on a mouse that has glycogen storage disease, that has a heart arrhythmia, and when that mouse gets really scared, it actually just falls over and dies. Its heart just stops. And so these diseases can be incredibly serious. So like I said, we know a lot about the genetic mutations within the genes that cause glycogen storage diseases, but sequencing all these genes can't always predict what those genetic causes are. We can't always find mutations in these genes, which is really weird because, that's the only way to get a glycogen storage disease, right? So that begs the question, what are we missing? So can you type in the chat what percentage of the genome actually codes for genes? And how much of it is non-coding or junk DNA? $132, 15%, great. Any other guesses? Yeah, So this is actually really surprising. Yeah, these are all really great guesses. But actually only about 1% of the human genome actually codes for a protein that has some function in your body. And that's pretty wild. That's like a pretty new discovery. When I was your age, 10, 12 years ago, I would go to my science classes and I would go to science museums and they'd say, 99% of the human genome, it's junk DNA. It's an evolutionary byproduct. It doesn't really mean very much or anything. But what we've learned in the last 10 years is that 99% of the genome actually regulates those genes and tells them when to turn on or off. And so in our recipe book analogy, let's say this really great recipe book, not only does it have all of these recipes and ingredients and instructions, it also tells you, when to make all of your recipes. It says, hey, this watermelon salad is really, really great for a summer picnic with your friends. Or it tells you, you should make this pumpkin pie when your family comes over for Thanksgiving. And let's say that there are issues in those instructions in the recipe book. You might not know that you're supposed to make pumpkin pie when your family comes over for Thanksgiving, and there's going to be no pumpkin pie. And if that pumpkin pie is actually a really important protein and it doesn't get made, you could have a really serious disease. And it could be just like there's a super bad mistake in the recipe for that pumpkin pie or that chocolate chip cookie or whatever protein or recipe you're trying to make. And so what I'm studying is how glycogen storage diseases might be caused by issues in these instructions on when genes are made or how much they're made. And those would be mutations in those non-coding parts of the genome that scientists aren't super good at interpreting yet. So how I'm studying this is, instead of looking at the DNA, I'm actually looking at the RNA. So if there are issues in the genome in that non-coding region that tell this gene to not turn on, or don't tell it to turn on, or don't tell it to turn on enough, you'll see reduced levels of RNA. And so if we look at how much RNA glycogen storage disease patients have for those glycogen storage genes, we would think that if this hypothesis is correct, we're going to see less RNA or even no RNA for those genes in the glycogen storage disease patients, and that would explain how they have the disease. And this is why this non-coding part of the genome, we like to call it a genetic switch because it turns those genes on and off. So why is this important? Like I said, right now we can only diagnose about 50% of genetic disease patients by sequencing their genomes. And if we want to improve that number, we really need to know which mutations to look for. So if I can figure out, hey, what are the non-coding mutations that are actually causing the body to not make RNA for these genes? If we can add those mutations to the list of mutations that doctors look for in patients, then they could be diagnosed really early. And early diagnosis is super important for improving medical treatment, knowing what treatments to give a patient. If you sequence a baby when they're born, you could find out that they're going to start getting symptoms of a glycogen storage disease in a year or 2 years. And you can start treating them even before their symptoms start, when you would normally discover that they have a disease. And that's really important because once your symptoms start, your organs are already getting incredibly damaged, and that's often irreversible. So if we can start treatment right when people are born or really early in life, that could really significantly help their health. Other ways it can help is that it can help patients know if they have children, what are the risks for their child. If they know that they have a mutation that's autosomal recessive, they'll know that their kids have a 25% chance of inheriting their disease if their partner is also a carrier for the disease. And that can help them plan whether they want to have children. It can help them plan whether to genetically test their children as soon as they're born. so that they can start treatment early. These are all things that are really helpful. And kind of most of all, not knowing why you're sick, it's really, really hard on families. Actually, my younger brother, when he was born, he had a rare disease that was incredibly severe. He was bleeding. He was getting like a lot of infections, and we didn't really know why for a long time until he got diagnosed. And that was really hard on my family. My parents didn't know whether they could safely have another kid that they wanted. And so when we finally got that diagnosis, it was really a great weight off of our chests. We knew how we could treat my brother and what options were available to our family. And so hopefully through my research, we can understand how to find these non-coding genetic causes of disease and help diagnose more patients and improve all of these outcomes for eventually all rare disease patients. So does anyone have questions? I know you guys put some together the other day, but if you have any right now about any of my research, about being a scientist, about going to college, studying science, anything. Yeah.
Oh, okay, I have one here from my classroom.
Okay.
So this child wants to know, the student wants to know if, like, obviously you had to go to school for many years, and yeah, it was a lot of time and effort. And do you, like, is it rewarding for you? Is that what you meant? Like, is what you're doing to you fulfilling, so that it was worth all of the time and effort and probably expense that went into your, it's your education?
Yeah, so there are a couple of answers to that. The first one is, yeah, it definitely took a lot of years, but I was doing something that I really loved, and I know that my research is going to help people. It's going to diagnose real patients, and it's going to help those families. And that is, especially considering my experience with my brother having rare disease, it couldn't be more rewarding. The other thing is that a lot of people don't know this, but getting a PhD, you have to pay for going to college most of the time if you don't get a scholarship. PhDs in science, not only is it free, they actually pay you to go to do a PhD and do research. So I actually make as much money right now as I did after college when I was going to work.
Some very surprised students and teacher in the classroom right now.
Yeah, it's a really great deal. It's hard work, but they pay us for it, which is super helpful. And, potentially, if you have a PhD, you can make a lot more money on the back end than with just a high school degree or a bachelor's degree. Okay, from Emmy, do people with this disease need a feeding tube? Not usually. So for most of these diseases, since we know a lot about the genes that cause them, we can optimize their diets to help them kind of stave off some of the worst symptoms of the disease. They don't really need a feeding tube. Often they will need to eat more often than you or I would, just because their blood sugar gets low really easily because they can't, like we talked about, they can't make glucose from glycogen. So their blood sugar gets low and they just have to eat more often because they can't store energy like we can. After college, are you going to continue with this career, or are you going to go back to music? I really love music. I play in an orchestra just for fun, but I'm going to continue with genetics just because I've really fallen in love with it. But hopefully I'll always continue to play violin. Actually, in my free time, I'm a wedding musician on the weekends. So maybe as a side gig. What techniques allow you to see mutations, and what role does computer modeling play? Ooh, that's an interesting question. So the techniques that allow us to see mutations, that's kind of what I was talking about, genetic sequencing. So we have all of these nucleotides, the A's, the T's, the C's, and G's that make up our human genome. And there are ways to figure out what order those A's, T's, C's, and G's are. There are a lot of ways to do that. Right now, usually what happens is you put it in a machine, and when it reads an A, it'll like flash red. And when it reads a C, it'll flash yellow. And a T and G, I think, are like blue and green or blue and black or something like that. And so when you see the order of the flashes of light, you can read the sequence of the gene. Computer modeling, it doesn't really play a role in sequencing, I would say, but there are a lot of computational models right now of how are these non-coding parts of the genome regulating the gene. It's just a ton of information. The human genome, I think, is 3 billion nucleotides, and that's just too much for any one person to comprehend. And so sometimes what scientists will do is they'll put that information into a computer and say, hey, computer, you can deal with all of this information, pull out what's important, and tell me what I should be looking at more closely. How long have I been studying this exact disease? Not very long at all, actually. So I'm a second-year grad student, and usually our first year, we work in a few different labs to kind of figure out what we're really interested in working on during our PhD. So I've actually only been working on glycogen storage diseases for a few months, since May.
What were you working on before?
So I worked a little bit between college and grad school on the genetic causes of diabetes. So while we think that diabetes and obesity are, caused by lifestyle, so, eating too much sugar, actually, if your parents have diabetes, even if you're not raised by them, even if you're in a totally different environment, you have an incredibly increased risk of getting diabetes. And so that tells us that there are some genetic causes of diabetes, of obesity, of all these things. And so I was trying to understand, what are those genetic causes? Can you genetically modify a baby before they're born to make sure they don't have any bad genes? Yeah, that's a really controversial one. So maybe a year or two ago, somebody actually did that in China. Right now, our gene editing technologies have a lot of problems. And they're, you can fix a gene in a baby, but these gene editing technologies, they make a lot of mistakes. And so they can cause other mutations in other parts of the genome. And those mutations could cause other diseases. So right now it's really risky to do that. And it's not really allowed for you to do it on babies before they're born. But there are other treatments called gene therapies where, once kids are born or after they're an adult, they can actually put the correct version of a gene into a virus and infect your body or infect the right parts of your body. And those viruses will insert that good copy of the gene into your cells so that now you have a functional gene product and a functional protein, and you can be healthy because you have a good copy of that gene. Have I seen the movie Gattaca? Yeah, it's a great movie and really scary. Just because now we're learning more and more about, what causes disease, and we can start to predict from someone's genome, you're in the top 5% of people that are likely to have a heart attack. And so there are a lot of ways that kind of information can be abused if it falls into the wrong hands. But of course, lots of ways that it can be used for great reasons. So if you know that you're really genetically likely to have a heart attack, maybe that'll convince you to eat really healthy and exercise a lot. We have a Socratic seminar on genetic engineering next week. Maybe we can view Gattaca. Yeah, I actually watched Gattaca in my 9th grade biology class. Any other questions? We can go over some of the ones that you submitted earlier. Yeah, how do you find patients to investigate if this is rare? Do you work with hospitals all over the country? I'm really lucky to work at Duke because we have one of the best experts on glycogen storage diseases here at Duke. And so patients come to see her from all over North Carolina and all over the country to see if she can help them. And so from that connection, my research advisor knows her really well and they work together a lot. And so that means that I get to look at a lot of different patients and sequence their blood for DNA and RNA and try and figure out what's going on. What was my favorite thing about college? Ooh, that's a hard one. I loved college. I would say that there are just so many options to figure out what you want to do with your life and what you're good at, and what really inspires you. Like I said, I came into college as a music major, but I went to UNC, which is a really big research university. And I just happened to see a flyer for some lab that was working on brain stimulation. And I thought, wow, that's super cool. And just kind of on a whim, I started working in a biology lab and realized that that was actually what I wanted to do with my life. And there are so many opportunities, especially if you go to a big school like UNC or like Duke. There are so many different majors. There are so many different student organizations. I got a lot of teaching experience. I taught violin through a student organization, and I realized that I really like teaching and doing research in the biology lab and teaching violin. I realized I kind of want to do both, and that's what inspires me to become a professor and do both research and teaching at a university like UNC or Duke in the future. Do I work in a team, and if so, how big? Yeah, definitely I work in a team. So I'm not a doctor, and when you're working with patients, obviously you need doctors. So we have lots and lots of doctors and genetic counselors that work, and nurses that work with the patients directly, and then help me get the samples and research them a little bit more. So there are probably like 5 doctors and nurses that I work with. I work in a lab that studies gene regulation. And that's like about 15 people. Not all of them work on my project, but they all work on something related. And so they all have really great advice and help that they can give me on what I'm studying. And then just in general, I go to talks and seminars from scientists that are around the country and even around the world, and get ideas from them for the problems that I'm trying to solve in my own research. How long have I been playing the violin? I've been playing the violin since I was 10 years old, so that's 15 years. Quite a long time. A good investment. You get paid a lot for being a wedding musician, guys. What is a non-science class or academic discipline that helped you as a scientist? I would honestly say that being a musician has helped me be a scientist as much as my science classes, because to be a musician in an orchestra, in a small group, you have to learn how to work in teams. You have to learn how to bounce ideas off of each other and compromise on what you're going to do in the end with your piece of music. And I think that has a lot of parallels to being a scientist. You have to be really open-minded and listen to other people's ideas on how to solve your scientific questions and answer them. And you have to be willing to work with the people in your lab and with the people collaborating with you to figure out, what's important to research? What's the best way to research those questions? I think that aspect of teamwork that's really intrinsic to learning how to be a musician are incredibly translatable to being a scientist. But also math. Math is important, guys. I didn't know this when I was in high school. Let's see. What are missense and nonsense mutations? So a missense mutation is you have a gene and there's just like one base pair. Instead of an A, it's a G. And that can cause really small problems, like it could just change like one little part of that RNA and one little part of that protein and not make a huge difference at all. Or it could make a really big difference. So let's say, let's say 1 egg versus 10 eggs in your chocolate chip cookie. That's like a really tiny difference in, the code of what's said in the instructions. But a really big difference in the end. If it says 1.1 eggs instead of 1 egg, probably won't make a huge difference to your cookies. They'll still be really great. So missense mutations can have a lot of different effects. Nonsense mutations are when the DNA tells the RNA to just like, it basically stops making the RNA early before the protein is supposed to end. And so that could mean that, you know, you're trying to make a pumpkin pie, and after it says cook your pumpkins, it just stops. And you just have pumpkins instead of pumpkin pie. And, you know, you just have a totally non-functional form of your protein.
I think the big thing they wanted to know is like the treatment for glycogen storage disease. And you covered just by changing diet, how that can really help the patients in that way, correct?
Yeah, I can cover a little bit. My focus is on the genetics and not as much the clinical symptoms, but I can tell you what I know. It really depends on, here we go. We have this really complicated pathway for how glycogen is stored and processed in the body. And so the treatment really depends on which gene you have mutations in. And so for some of these, let me find glycogen storage disease 2, that can be treated by gene therapy, which is what I talked to you about, you know, you can put a functional form of the gene into your body. That's the treatment for that one. Others of these are a little bit less severe, and by changing the amount of carbohydrates or proteins you eat, that can help you really manage your symptoms a lot. I'm trying to think. Others, these are all different enzymes. So sometimes you can actually just treat patients with the enzyme itself, and it'll help them process glycogen, like in the same way that that would happen if their cells just made it as a part of their healthy function. So
like insulin and diabetes, like that sort of treatment. Okay.
Exactly. Yeah, but we're always discovering new treatments. As gene editing technologies improve, there's a lot of potential for gene therapy for all of these diseases and potentially all genetic diseases. And so that's really exciting and something that we should definitely be out on the watch for.
Thank you so much for this. It's fascinating.
Yeah, you're welcome.
And yeah, it was engaging because you also included like your information about college and some background in addition to just the genetics of this. So thank you.
Yeah, you're welcome.